And so the genetic pathways that regulate entry into dower over reproductive development have been well studied. Dower was discovered in the 1970s when people first started working with worms. And since then, they've been trying to figure out how the worm knows to enter into dower, what controls it, and they have exhausted that question. And they have come up with a lot of different pathways that regulate dower entry, but the most integral and the most important is the daf-2 pathway. And what they have come to understand from studying the daf-2 pathway further is that it also can play a role in lifespan of the worms. And so the first important gene that controls both dower and longevity in worms is the dower-forming gene 2, or daf-2 gene, which encodes the daf-2 protein. And daf-2 protein is a homolog of an insulin or IGF, or insulin-like growth factor, like receptor. And so this is a transmembrane receptor, or a receptor that goes through the cell membrane and senses pro-growth signals like insulins and IGF, which is a growth factor, and then responds accordingly. Additionally, inside of the daf-2 pathway, another gene that was identified to regulate dower and lifespan is a gene called age1. It was named so because it has an aging phenotype, super creative. And the age1 protein that's encoded by age1 gene is a phosphatidylinositol 3 kinase, or a PI3 kinase. And this is a kinase, or an enzyme that phosphorylates PIP2 to turn it into PIP3. And we don't need to know the exact downstream targets of this PI3 kinase, but what you need to know is that this first phosphorylation step of PIP2 to PIP3 being phosphorylated starts a phosphorylation cascade or signaling cascade within the cell that can ultimately phosphorylate some downstream targets that affect transcription, one of which is daf-16. And daf-16 is the final member of the daf-2 pathway that you need to know. The daf-16, or dower-forming 16 gene, encodes daf-16 protein, which is a transcription factor. And as we talked about in the previous lectures, transcription factors are proteins that affect transcription. They might enhance it, they might suppress it. In the case of daf-16, it's a repressor, so it turns off genes. And it's similar or homologous to the mammalian FOXO transcription factors. And what daf-16's ultimate function is to repress transcription of genes that are involved in normal growth and development. And so we're going to put these 3 genes together into the pathway and show you what happens under normal conditions, right? And so under normal conditions, when environment is favorable, there's not a lot of worms around, there's a lot of food, everything is good, there are neurons that can actually sense the environment to figure that out. Those neurons will secrete or release daf-2 ligands, pro-growth signals, that will bind to the daf-2 receptor, right? So daf-2 is transmembrane receptor, the ligand binds on the outside or the extracellular side, and then daf-2 can transmit a signal to the inside of cells. And when it does that, and when daf-2 ligand binds, it activates daf-2 to in turn activate that PI3 kinase age1. And when age1 is activated, it can do what kinases do, which is phosphorylate. And it starts a phosphorylation cascade, which ultimately leads to the phosphorylation of the transcription factor daf-16. And when daf-16 is phosphorylated, it's basically prevented from entering the nucleus because that phosphate group is highly charged and negative. This transcription factor with a phosphate group on it is unable to enter the nucleus. And so phosphorylated daf-16 remains in the cytosol. And their genes that it would normally repress are on. And so gene expression for reproduction and growth is on. The cells can reproduce and the worm can grow normally. And that makes sense, right? Because conditions are good. There is enough food, there is enough space, there is a low population of worms. So why not grow and reproduce? from the worm's perspective. Then when conditions are bad, the neurons can actually sense either too many worms around or not enough food. They will not produce the necessary pro-growth signals, and daf-2 will go unactivated, right? So it's still here, it's just now unable to activate the PI3 kinase age 1, which means there's no phosphorylation cascade, which means daf-16 at the end of that cascade is not phosphorylated. And now, since it's not phosphorylated, it can enter into the nucleus, and it can basically bind to the DNA, as transcription factors do, and turn off transcription of genes that would promote normal growth and development, which leads to the formation of dowers, or entry into dower. And so under bad conditions, daf-16 is in the nucleus and basically causes dower formation, which also makes sense, right? Because you would rather enter into this sort of hibernation state when stuff is bad rather than continue reproducing and growing. And so that's how the daf-2 pathway affects growth and reproduction, as well as dower entry. But daf-2 can also, kind of as a side effect, affect lifespan. And so there are loss of function mutations in daf-2, meaning that daf-2 does not work as well as it normally does, that have been pretty well studied. And you can actually see here a lifespan assay of several different genotypes of worms, wild type in circles, this daf-2 weak loss of function mutant in X's. And so the way that we read genes in C. elegans is this is the gene name, and in parentheses after it is the allele. And so this is that mutant loss of function allele in daf-2. And what we can see from the survival curves here is that compared to the wild type circles, daf-2, this weak loss of function mutation, has a much longer lifespan. And so this mutation can actually extend lifespan. Not only the average lifespan, as you can see here, but that kind of, but the overall lifespan as a whole, right? Because there are some worms with this daf-2 mutation that lived to almost 60 days, which is 2 months, which is 3 times their normal lifespan. And this is likely through that same pathway we just talked about in terms of dower, and where daf-16 is either in the nucleus or in the cytosol. And so under normal conditions and in wild-type worms, when we look at daf-16 here, which you can see in green, labeled with GFP, you can see that this is a picture of a worm, and daf-16 is diffuse looking. It fills up this whole worm's body. And that's because in wild-type worms, daf-16 is in the cytosol. These worms are well fed. There's no reason for daf-16 to be in the nucleus. It is, all the conditions are good. But under the same good conditions in the daf-2 mutants, you can see that there's now this punctate localization of daf-16. And those puncta are each nuclei of cells. And so just this mutation alone is enough to move daf-16 into the nucleus, which should trigger at least a little bit of entry into dower and repression of the genes involved in reproduction and development. And so that's one possible mechanism for how this loss of function mutation in daf-2 actually extends lifespan. It might do it by diverting resources away from reproduction and development, because daf-16 is sort of repressing the genes that would be necessary for that, not making the proteins you need. Those worms are not reproducing and developing as they normally would. And we know that's one of the big kind of age-related issues, right, is a shift of resources from the soma to the germline. And in this case of daf-2 mutants, there might not be as many resources taken up by the germline, and so the soma and the rest of the body can actually survive to live longer.